So we're presenting our data today in order to identify potential molecular mechanism of resistance to immune checkpoint inhibitor in MSI-H colorectal cancer patients. We know these drugs are approved checkpoint inhibitors which show very promising efficacy in first line for this patient population, but up to 30% do not have any benefit. So we wanted to figure out, are there potentially innate mechanism of resistance to immune checkpoint inhibitor in MSI-H tumors? We went into the CARIS database of 25,000 colon cancer patients and identified about 1,500 with MSI-H tumors. Then we looked what patient population received immune checkpoint inhibitor in first-line treatment. And there were about 700. From this 700, no, actually 238 exactly. From this 238, we checked what patients received more than 6 months of treatment or less 6 months of treatment as an indication for response to treatment. We did that, and the survival curves between both, as expected, was incredibly big. Then we looked for the transcriptome signatures because we know that certain innate mechanisms of resistance include beta-2 microglobulin or loss of HLA. So our hypothesis was to look and characterize the gene expression of HLA genes, the MHC-I and the MHC-II classes. We did the gene expression and identified that a signature in MHC-II predicted the response to treatment. Now, we also looked further what that means if you have higher expression of the MHC-II class gene expression, and showed that this score, when it was high, was associated with certain molecular genetic alterations, but also with immune cell infiltrates. So it's a T cell inflammatory signature. It's associated with CD8, B cells, NK, M1 and M2 macrophage infiltration. So there is some really good understanding rationale why these tumors seem to do much better, because you have all the immune cells in the tumor in an inflammatory stage, so that the immune checkpoint inhibitors can actually have a significant benefit. So I think this is the first insight where we understand that the expression level of HLA genes, particularly the MHC-II class, may play a big role in innate resistance.